So here we go with the last video I'm going to produce for this course, in which I want to deal with a group called the Antliophora, which comprises the last, let's say the last orders of the holometabola. Whether there's two or three, as you'll see, has become an issue of some dispute, or at least alternative proposals, let's say. So the Antliophora. The name actually refers to a pump, so a pump-bearing group of organisms. Supposedly, when this first group was first proposed, one of the synapomorphies that united the orders Mecoptera, Diptera, and Siphonaptera was the presence of a sperm pump in the male. So in the males of this group, they don't produce spermatophores like many other insect groups do. That is a package of sperm, a glandular secretion containing sperm. They have free sperm that they transmit to the females. So there's no spermatophore. Even though it was proposed as a synapomorphic character for all of these orders, in fact, a sperm pump is absent from a good number of Mecoptera, perhaps all. Not all of them have been examined in detail. And it's also absent from a big chunk of the true flies, the Culicomorpha, the group that includes both, not both, but mosquitoes and midges and similar creatures. And then we also find a sperm pump present in Strepsiptera. So you remember back to our discussion of long branch attraction in the Strepsiptera problem, is that what the proposal by Whiting was that the Strepsiptera are most closely related to the Diptera, and they suggested in their paper that the presence of a sperm pump was a synapomorphy, putting the Strepsiptera with the Diptera here in the Antliophora. Well, that idea has been shot down quite convincingly. One of the main things being is that the sperm pump of a Strepsipteran male has no cuticular parts, whereas that of the Antliophora does. Even so, it seems to be clear from the morphological evidence that the sperm pump in Antliophora is not homologous across groups either. So the supposed one synapomorphy that united all this group is now pretty much debunked as a characteristic, and that it's apparently convergently evolved in fleas, in flies, and things like that. Nevertheless, even though that morphological underpinning for this group has been discredited, the monophyly of these three orders is strongly supported by all the molecular evidence. So we're kind of stuck with a name. So even though they don't all bear a pump, or at least don't all bear the same sperm pump, we are going to treat them as a monophyletic group. So what the heck is this sperm pump that I'm talking about? So this is a paper that was looking at the structure of the sperm pump in Strepsiptera and in flies and fleas, so Diptera and Siphonaptera. And the illustration I have here is of the sperm pump in a crane fly. So this is in the order Diptera. On the left-hand side here, we have the histological sections of the tail end of a male crane fly, and then on the right here the interpretation of that in the form of a line drawing. So basically this corresponds to this structure here, which is the endophallus, or the phallic structure. Actually, it's a combination of individual elements, but that's the intromittent organ of the male. Up here we have the testes, and then the ducts leading down here, where this is the vasa deferentia, and the ejaculatory duct is somewhere here as well. You can see it in this part here. So basically the idea of what a sperm pump does is so that we have the vasa deferentia here, which connect to the testes. So the sperm are coming down this way, and then there's this chamber here, and an ejaculatory apodeme here. So this area in white is a cuticular structure, sclerotized cuticular structure, and there's a pair of muscles labeled M and big muscle here labeled N that basically pull on this ejaculatory epidyme, and pulling it one way opens the this chamber, creates low pressure, and the sperm flow in, and the other way it expels them through the through this canal right here, labeled EPHT. So that's what I mean by a sperm pump. Although the the structures that are found in fleas and flies appear to be convergently evolved. They have lost the spermatophore, maybe that's the underlying synapomorphy, and they have evolved mechanisms to assist in the propulsion of these free sperm through the ejaculatory duct into the female's gonopore. So let's start out with the order Mecoptera. There's lots of common names for the Mecoptera because they're such a strange little group that they kind of inspire common names. Scorpion flies, hanging flies, earwig flies, snow scorpion flies, and there are even more groups besides that. When I used to teach insect morphology, using Mecoptera as exemplars for understanding the external anatomy of insects was a very good choice because they have very generalized morphology. They have typical mandibulate mouth parts, so they're chewing, and all the pieces are there and are very easily recognizable. They have two pairs of, I'm calling it here, homonymous wings. By that, all I mean is that the forewings are very similar in size, shape, venation to the hind wings. So they're all very similar, homonymous. They have generalized thorax and venation, so you can recognize all the fundamental pieces of a pterothoracic segment. Sometimes when you get in these specialized groups, that the thorax itself becomes very specialized, and one part gets hugely overgrown and others get highly reduced, and so it's hard to recognize. But in scorpion flies, they're very easy to recognize. They have eruciform larvae. By that I mean caterpillar-like larvae. And many Mecoptera have what's called a rostrum. Basically, they have the head where the mouth parts are prolonged into kind of a beak, and this is formed by an extension of the clipea. So I'll have an illustration for you in a second to explain what I mean by that. But don't jump to the conclusion that all Mecopterans have this rostrum, because they don't. Some have very, very typical looking, more or less hemispherical heads. This group is very common in the fossil record. It dates back as far as the Permian, and there were lots of species. But today is really more or less a relict group with only about 600 species still living around the world. So here I want to illustrate what I meant by the rostrum and its extension. So ignore these three images here at the bottom. These are the Diptera. We'll return to those when we get to that order. But up here we have an example of a member of the family Panorpidie, one of the main families of Mecoptera. And the mouth parts are color-coded: labrum, mandibles, clipeus, laciniae of the maxillae, labia, hypopharynx, and then the maxillary palps. And you can see that this elongate beak of this scorpion fly is composed primarily of the clipeus, which has become extended. If you look at, that's an anterior view. If you look at a posterior view, see the labium is not that large. The mouth parts are born at the tip here. The maxillae and maxillae and mandibles are more or less typical in shape, and the labrum is fairly small. That's the yellow part here. But it's this elongate purple clipeus that forms the basis of the rostrum in Mecoptera. And let's go from this drawing to look at a real one. So here we have a representative male of the family's Panorpidie. So here's the mouth parts down here. You can see the maxillary and labial palpi, and this in here it's kind of orange. The color of that is mostly the clipeus that has become extended and bears the mouth parts at the tip. You can understand how these creatures get the common name scorpion flies because the male genitalia have this large bulbous section here, and it's kind of held recurved over the dorsum of the abdomen, looking like a scorpion sting. But this obviously cannot sting at all. These are copulatory apparatus and not a defensive mechanism or an offensive mechanism at all. These are perfectly harmless creatures. In the Mecoptera, there's only 9 living families around the world, so it's depauperate both in species and in families, and many of those families have very, very few species. In fact, 95% of the 600 species that occur in the order are found in only 2 families. These are the scorpion flies, the family Panorpidae, and the hanging flies, the family Betacidae. Both of these are very common here in our Ohio fauna. The scorpion flies, as in their immature stages, well, actually also in their adult stages as well, are basically scavengers. They're looking for dead arthropods, or maybe they'll feed on dead snails and worms as well, but they're basically scavengers. While the betacids, or the hanging flies, are predatory. And the betacids, as their name implies, hang upon vegetation, dangling their legs downward. And they have a large modified pretarsal claw on the apex of their hind legs, which they use to snag insects and other arthropods, like spiders or something like that, snag them out of the air. I've showed you a male panorpid. This is a female panorpid. They don't have an appendicular ovipositor. Basically, they just have a modification of the apical abdominal segment. So it's kind of a telescoping tube that comes out for them to lay eggs. You can see Circe here. What else can we see on this creature? The elongate rostrum again. So it's very easy to tell males from females. This family is very common in moist woodlands, and you'll see them perched on low herbaceous vegetation in these deciduous forests. As I said, very common here in central Ohio. This is a panorpid larva, a ruciform, so caterpillar-like. It does have prolegs back here. These are also scavengers. If you want to grow these, you can feed them dead insects or worms and things. You can see they have well-developed compound eyes and well-developed antennae here as well. This is a betacid, or a hanging fly. As I said, they basically hold on to the vegetation, and they'll dangle these hind legs downward. And you can see these enlarged pretarsal claws. People call these tarsal claws, but they're actually from the pretarsal segment. And they use these to snag insects to feed on. So these are predators, catching things on the wing. They kind of look like crane flies. I think you might have jumped to that conclusion, but clearly note that they have 4 wings. And when I said homonymous, you can see how they're all more or less the same size and shape. Well, that comprises, what did I say, 95% of the species of Mecoptera. There's some really interesting things. Both betacids and panorpids are very common here in central Ohio, but there's another family that is actually found in the not the Alleghenies, not the Adirondacks. What do you call that mountain chain? Oh, the Appalachians. Too many A's. The Appalachian Mountains. So if you go a little bit farther east from where we are here, and you're lucky, you may run into the family Panorpodidae, which are, sometimes people call them short-faced scorpion flies. You can see here in this anterior view that the head is circular in frontal view. They don't have this elongated rostrum. These are found not only in eastern North America, but also in eastern China. So they have a peculiar distribution, I think almost undoubtedly representing extinction over the many years that they've been in existence. One other common interesting panorpodid is some of them have shortened wings, so they cannot fly. They're brachypterous. As a matter of fact, this genus is called Brachypanorpa, referring to the short wings, although this species has fully developed, and this sex has fully developed wings. One of the other families that is here in central Ohio is the family Boreadeae, which go by the common name snow scorpion flies, because it's not uncommon that you'll find that the adults are active during the winter. So if you get a nice bright sunny day where it warms up a little bit, you can find these things crawling around on the snow. Both the adults and larvae feed on mosses, so they're characteristic of moist forests Um, so they're feeding on the mosses that are growing on the rocks, on the trees and things like that. Um, Both sexes have reduced wings that in the males actually function as claspers. They use them to grasp the females during copulation. Um, I guess that's ocelli lost, rarely redeveloped is a little morphological trivia point. These are very distinctive things. Um, their phylogenetic position has been in some dispute. Willmann uh, was one of the people who was really focused on studying Mecoptera in the 1980s or so, on the basis of morphology, obviously at that time period, and he placed the Boreads as a relatively apical group within the phylogenetic tree of the Mecoptera. And in fact, there was some suggestion that perhaps they were closely related to the fleas. We'll revisit that point a little bit later. But more recent work based on molecules, the One Kite project, and then a paper published just last year, also based on transcriptomes, placed the Boreae as the basalmost, the first branch off the phylogenetic tree of the Mecoptera. So there's some disagreement about where they belong, although if you believe the preponderance of evidence is coming out of the molecules, and you're going to put these at the base of the tree. This is a female Boread, so here she is standing on mosses. Some of the species, and including one of our local species, have a distinct metallic tint to their body. Remember, these are wingless, or at least highly brachypterous, and the females here are wingless. They do have an elongated rostrum. I can tell it's a female because she's got this extended apical segments to function as an ovipositor, and also you don't see the male wings are about this long, and they're kind of hook-like to grab onto the female during mating. Two other groups I want to mention in the context of Mecoptera. So what do we have? We have the Pronorpids, we have the Panorpodids, the Batassids, the Boreas. So that's 4 families. I'm going to talk about 2 more. That brings us up to 6 families. And then the other families are all, none of them are found in the U.S. The Choristids and the Austropanorpids are found in Australia. So the first one I have listed here, it is not found in North America. This is the Nanochoristidae. I bring them up for a couple of reasons. The first is that in Willmann's work, he placed the Nanochoristids as the basalmost group of Mecoptera. Remember, this is the position at which the modern molecular evidence points to the Boreads being there. Modern molecular evidence basically switches the positions of these 2 families, Nanochoristids versus Boreads. Nanochoristids are interesting because they have that transantarctic distribution. I believe I've mentioned this before. This is when you have things that are found in like southern Chile and in southern Argentina, and then then they're found in New Zealand and in Australia. So this is the distribution that the Nanochoristids have. They're interesting to me. I find them very interesting because they have aquatic larvae. This used to make sense if you put them at the base of Mecoptera, because we have numerous groups that have aquatic larvae at the base of the tree. We have all the Trichoptera. We have the basal group of the Neuroptera with aquatic larvae. We have the Megaloptera that are aquatic. We have the Hydrodephaga in the beetles that are aquatic. But the aquatic larvae of Nanochoristids, if you believe the newer evidence, is, well, we don't know that it's apomorphic or convergently derived itself. Maybe it's a holdover from old times. And there's a reason that we don't know the answer to that. And that gets me into the next family, the Meropiidae. The Meropiidae are also known as earwig flies, and you'll be able to clearly see why in just a second if you didn't see them in the lab. This is really a very relict group. There's only 3 species known of Meropiidae. uh, in two genera. One genus is the genus Austromorope. Austromorope was originally only known from Western Australia, and in fact is very, very rare in Western Australia. But then just a few years back, 4 or 5 years ago, a species of Austromorope was discovered in all places in southeastern Brazil, in the rainforest along that part of the continent. So not the Amazon rainforest, but the Atlantic rainforest along the coast of Brazil. So we have a really disjunct distribution in this species. The other species of meropiids is the genus Merope, which has a single species, Merope tuber, which is endemic to the eastern United States. It might, I don't recall, I think it might sneak up into southern Canada, but it goes west as far as Arkansas, Missouri, Oklahoma, that area. We actually have, Merope is not uncommon here in central Ohio. You can expect Merope tuber to come out. The adults are nocturnal, so they fly at night. They'll be coming out in July, August, and continue coming out until you really get a hard frost at the beginning of winter. I guess 2 points to make here. If the genus is Merope, why do we call the family Meropiidae instead of Meropidae? Well, that's simply because there's another genus. It's birds that have already used the family, the genus Merops, which I believe are bee eaters, have kind of glommed onto the name Meropidae. So the entomologists had to go for Meropiidae in order to have a family name for this distinct group. The other thing, and this is what really excited my major professor when I was a graduate student, was that even though Merope tuber has been known a long time, it was described in 1833, so a long time ago. It's been known for almost 200 years. But the larva of Merope tuber has never yet been discovered. Merope tuber seems to be associated somehow with intermittent streams, the presence of water. It seems not to be truly aquatic, but you collect adults around these areas. My major professor had a well in his backyard, and during the summer when he'd have parties and the lights out, occasionally a Merope would fly in, and he'd jump up and down and be all happy for having collected them. So, as I said, while we can collect adults, I've collected them live and brought them into the lab, and they don't seem to eat anything. They don't seem to do anything. They just seem to sit there for days and days and then die. So nobody really knows what the biology of this creature is. This is one of these fascinating little tidbits of a problem that would be cool to figure out what exactly it does. I suspect that the larva probably looks a lot like a beetle larva, and perhaps it's already been collected, and just that people have misidentified it as a beetle larva. Let's take a look at some pictures. So this is a nanochoristid. Again, no rostrum here. A very nondescript small creature, if you ask me. As the name implies, they are small. They're only 5-6 millimeters long. The family Choristidae is basically the panorpids of Australia, and so that's where you find them. They're very common, about that size. These guys are much smaller. So that's what the adult looks like. The larvae, I couldn't find a good photograph of the larva, but you can see they're kind of that same eruciform larva that I showed you with the panorpid, the scorpion fly. Well-developed thoracic legs, nicely developed head capsule, but they are aquatic. So again, wouldn't it be nice to discover the larva of Merope tuber so we can start to understand how the life habits of the larvae of Mecoptera have evolved. This is Merope tuber, and you can see why it's called the earwig fly. This is a male, and these are the claspers at its tail end. So while a panorpid, the scorpion fly, has that bulbous genital apparatus that it holds back over its neck, Merope tuber has these big elongate claspers. I mean, it's segmented, and so this arm can move in relation to the other arm about this axis, and so presumably they're using these to clasp the female. As far as I know, no one has actually observed males and females mating, so that's a bit of a presumption about how these structures are used. This is a female Merope tuber. This is actually one that I caught and had brought into the lab and took some pictures of it. Kind of nondescript. The females are kind of nondescript, kind of looking like little, almost like little cockroaches, right? As I said, these come out July, August, September. We would catch them. We had a Malaise trap down on the Hocking Hills. We'd get about 1 or 2 every week in that single Malaise trap. So they're not uncommon. Over a couple of years, we collected more than 100 of these things. I'm sure we didn't extirpate the population, so don't worry about that. We kept the traps going literally all year round. And toward the end of the year, I actually had one trap catch in which we got a Merope, a Meropeid, a Boreid, a Batacid, and a Panorpid. So 4 families of Mecoptera, all in the same trap catch in 1 week. This is the Western Australian species, Austermerope. This is Poltoni. You can see again the same elongate claspers. Obviously, it's the same family, but it's found literally half a world away. And then this is the South American species. It just makes you scratch your head about it and wonder perhaps there are more of these species around if we just collect in the right time, place, and use the right method for them. So that's basically all I want to say about Mecoptera. And move on to the fleas. And I'm really not going to spend a heck of a long time here with the fleas either. I'm sure fleas are familiar to almost everybody. Has somebody never seen a flea? All fleas are, well, I guess I can start out by saying fleas are a very compact group in the sense of variation in their morphology, their biology. Most fleas are pretty much alike. Morphologically, they're laterally compressed because they live on the bodies. They're all of mammals and birds, primarily mammals, as you can see from that number there. 94% of the species of fleas are ectoparasites of mammals, and so they have this flattened body shape to be able to slide to get between the hairs of the mammals or between the feathers of a bird. Of those 94% of fleas that are parasites of mammals, 75% of them are found only on rodents. So it's really a group that has just taken off and gone crazy, living on rodents. All fleas are apterous. They have no wings. They have reduced, I should probably put a semicolon there because the adjective reduced here refers to the eyes, antennae, and thorax. So without wings, the thorax becomes smaller. The eyes are small. The antennae are barely recognizable as antennae. The adults are blood feeders, and they feed upon blood using a beak that's formed from the, oh my typos, labrum, L-A-B-R-U-M, and the maxillary laciniae. So unlike the Lepidoptera, in which it's the galea of the maxilla that forms the proboscis. In the fleas, it's the laciniae, that other lobe at the apex of the maxilla. So the adults are blood feeders. The larvae are not blood feeders. The larvae are not ectoparasites. These things are ectoparasitic only in the adult stage. The larvae basically feed on organic debris found in nests. So dander, hair, any organic debris in a nest. That's what the larvae are feeding on. So if you want to get rid of fleas, it's one thing to poison all of the fleas that live upon, let's say, your pet dog or your pet cat. But you have to recognize that the larvae are still there, living in the bedding that the animal might lie in, or in carpeting, or anything like that, where they can find enough nutrition to feed on. And so you'll have flea infestations continually emerge even after you've purged the adult animal, purged the adults from your animal pets. So this, I put it in this illustration just to show this degree of lateral compression of the body. So from side to side, the animal is compressed. From top to bottom, though, of course, they're quite elongate. They have they jump by means of the muscles in the legs here, particularly in the coccyx. And the flea legs are also quite well known for having these pads of that same elastic protein resilin that I mentioned in the context of the Lepidopteran proboscis. So the muscular contraction that deforms the legs, moves the legs into a spring position, can be released from this elastic protein, is stored in and then released from this elastic protein resilin. We have, let's see here, the eyes. Can't really see the antennae in these guys. These are palpi here. And this segment, and this segment are the three thoracic segments. So the rest of this is all abdomen here. So the reason I started out this video saying, well, we're going to talk about 2 or maybe 3 orders, is that the most recent molecular studies, phylogenomic molecular studies of the Antliophora is in Tihelka et al., published just last year. And they found, on the basis of transcriptomes, mitochondrial genomes, and they had another data set of aligned molecules. They found that the Siphonaptera here in orange are actually a group found within the larger Mecoptera. So in their Mecoptera, they had a Borea, the snow scorpion fly. They had a Panorpid. They had a Bidicus. They even had Nanochorista. And then in their outgroups, they have these are Lepidoptera, and then down here we have Trichoptera, and then a bunch of flies down here, which we'll get to in just a second. But basically, interpreting this cladogram tells you that fleas are just highly modified scorpion flies, modified for this ectoparasitic way of life. And therefore they propose, this paper proposed, that the Siphonaptera should not be continued to be treated as a valid order of the Holometabula. So just as we found that the termites, the Isoptera, are nothing more than specialized cockroaches, these results, which are consistent with the one kite results, come to a similar conclusion that the Siphonaptera are nothing more than highly modified Mecoptera, and so that we should not continue to recognize these as orders. So in their classification, they have one order Mecoptera and then another order Diptera, which I guess it's time to move on to right now. So Diptera, the true flies. It used to be that your textbook had the Hymenoptera as the last group of last group of insects that they treated, because Bohr and DeLong basically bought into that idea that the complex biology, eusociality that we find in Hymenoptera means they're the most evolved. But of course, evolution doesn't proceed in a ladder-like form, even though in a book you have to do things in one dimension from front to back. You can easily make the case that the true flies are really one of the highest developed orders of insects. You can make a very strong argument that they are the most ecologically diverse group of insects. We have aquatic forms, terrestrial forms, we have freshwater forms, we have marine forms, we have detritivores, scavengers, predators, feeding on just about almost, don't forget the herbivores, there's plenty of herbivores, fungivores, they're very diverse biologically. And they're also probably the most well-studied group of insects. Part of this is, well, I guess there's two two veins here that contribute to that. One is that many Diptera are important vectors of some pretty devastating human and veterinary diseases, veterinary for that point. Bill Gates famously named the mosquito as the most dangerous animal alive because of all of the people every year, hundreds of thousands of people die each and every year because of diseases transmitted to them by true flies. Things like malaria, dengue, leishmaniasis. There's a long list of diseases that true flies transmit. So they've been of great interest because of that, and also several species of flies, most prominently species in the genus Drosophila, have been used as model organisms to study genetics and development. So they are very well known. I think this is correct, that the first organism to have its genome sequenced was Drosophila melanogaster. It was the very first. And of course, since the early 1900s, Drosophila has been used for genetic studies and studies of development. There have been some notable dipterists that we've encountered, and whose influence has been felt well beyond the scope of the true flies themselves. Willy Hennig, the founder of phylogenetic systematics, was a dipterist, and I'm not sure if I've mentioned this guy before. Actually, a Swedish entomologist, René Malaise. Despite the name René, he was a Swede. He invented the Malaise trap, which has really revolutionized our concept of what insect diversity truly looks like, because this is a trap that is specialized for catching flying insects. There's 1,000,001 great stories about true flies, and I would really strongly recommend a recent book that's been published by the curator of Diptera at the Natural History Museum in London, Erica McAllister, called The Secret Life of Flies. And this is Erica. She's got a great sense of humor, and she is a regular attendee at the Entomological Society of America. So if you ever get a chance to hear her talk, don't miss it. She's very entertaining. And as I say, in terms of flies, there are 1,000,001 fascinating, sometimes troubling stories to go along with them. You can also follow Erica on Twitter. So, true flies. You're probably familiar with the basic characteristics that these organisms all have. If the adults have wings, and there are many true flies that are apterous, so that little caveat. If they have wings, then they only have really one pair of functional wings, functional in the sense of producing the aerodynamic forces that enable them to fly. They do have hind wings, but these hind wings are reduced into balancing sensory organs that we call halteres. In the true flies, the labial palps are fused to the apex of the labium. Some true flies still have mandibles. The bite of something like a horsefly comes from the razor sharp mandibles that they have, but the fusion of the labial palps to the labium forms a composite organ called the labellum, which is used to imbibe fluids by those. Let me just leave it at that. It's used to drink fluids. Diptera are in the adult stages are basically fluid feeders. The larval stages have mandibles, and of course they're feeding on a wide variety of substances, but as adults they are fluid feeders. So let's go back to this image, and same color coding here, but really where I want to focus your attention, this is an anisopodid, so this is a gnat. There's lots of gnats in terms of common names, but and here we can see this blue structure here is the composite, the fusion product of the labium and the labial palps to form the labellum. And over here in the Californias, this is a blowfly, your more typical fly. You see the labellum here with the micro channels that are used to facilitate the uptake of fluids. There is a labellum here in the mosquito, but this structure, as you're aware, is protective of the has a protective function for the stylets that the mosquito uses to take, in the case of a female, a blood meal, or in the case of both male and female, to take a meal of nectar or some other sugary fluid. So the labium and the labellum are protective and also sensory structures here. I put this illustration here to show you the halteres. So in this syrphid, in this flower fly, we have typical forewings, and then this yellow club-shaped structure here are the halteres. It's it's here on the other side. It's the same color as the middle leg, so it kind of blends in there. But you can see it clearly here on the fly's right side. Just like the forewings, these are beating up and down during flight, and they act as gyroscopes, such that when the fly the fly's flight path veers left or right, up or down, rolling, all of those, because these are beating, they have some inertia and don't immediately follow the path of flight, and so that puts a strain on the base of the haltere. And there are cuticular sensory structures here that sense that stress and communicate that to the fly, so that it can interpret and understand, well, an additional sensory input in addition to go along with these monstrous eyes that it's got up there, to understand how it's flying and to get in the direction it wants to go. Okay, so Diptera classification. A lot of the terms that are used to describe flies, well, not surprisingly, come from their classification. The issue is that a lot of these terms are obsolete in the sense that they don't refer to monophyletic groups. So I'm going to present here a traditional classification, because you're going to see these terms very often, and then we'll get into the modern conception of what the classification looks like that's reflected in the article you read. So traditionally, the flies, the entire order, was split into two groups. On the one hand, we had the Nematosera, and on the other hand, we had the Brachycera. If it helps you to remember, the CERA at the end here refers to, it's a Greek word, I believe it's Greek, that means horns. So this is commonly used to refer to the antennae. Horns, you know. But the Nematosera, the thread-like horned flies, have long antennae, multiple clearly distinguishable segments, whereas the Brachycera have very short antennae. I have a nice picture here to illustrate this coming up. Then once we are into the Brachycera, that is then subdivided into the Schizophora versus the Aschiza. So the ones who have a split head versus the ones that don't have a split. And this split refers to the frontal suture down here. So the Schizophora have the frontal suture, the Aschiza do not have a frontal suture. Within those that do, they were subdivided into the Orthorapha, or the straight seam, referring to the frontal suture, and the Cyclorapha, not really circular but arched frontal seam. And then finally, so let's step back here. Divide all of Diptera into Nematosera or Brachycera. Divide Brachycera into Schizophora or Aschiza. divides Schizophora into Orthorhynchus versus Cyclorhynchus flies, and then divide the Cyclorhyncha into calyptrates and acalyptrates. Again, the A prefix here means without, and so this is basically saying the flies with calypters and those without. And the calypters are large lobes on the trailing edge of the forewing. All right, let's illustrate some of this stuff so it makes a little bit better sense. On the left-hand side, we have a crane fly here belonging to what the old classification would call the Nematosera. And you can see these thread-like antennae with multiple clear segments. You know, this is clearly a typical segmented antennae, much like you saw in that picture of the female of Merope tuber. Whereas over here in a Brachyceran, we see that the antennae are very short, and the vast majority of it is basically three segments: the scape, the pedicel, and then this one big third segment. And from the surface of this third segment arises this arista. Go back many videos now to the Paleoptera when I showed you the antennae of mayflies and dragonflies, and I said they were aristate antennae. That was a reference to this structure in flies. So this almost thread-like, I guess that's not good because this is referred to as being thread-like, almost hair-like projection from this segment. This is actually the rest of the antenna that's become very highly reduced, and usually all that's fused together into one piece. So Nematosera versus Brachyptera. Long horns, short horns. So the classification that's developed in the last, I would say about last 20-25 years has recognized that a lot of that traditional classification, there was a lot of truth to it, but basically all of those pairs of terms I had, Nematosera, Brachycera, Schizophora, they identified true monophyletic groups on one half of those pairs, but the other half was a paraphyletic group. And so the actual phylogeny of the true flies, instead of being this dichotomous branching into equal parts, so equal subdivision into, let's try that again, subdivision into equal halves at each split, is actually more like a ladder-like chipping off of a piece as we go along. And so many of those groups, Brachycera is a monophyletic group, Schizophora is a monophyletic group, Cyclorhyncha is a monophyletic group, but they're the ones with which they're paired often are not, or it's not clear that they're monophyletic groups. The other thing I want to point out is that the Diptera systematis had made extensive use of an additional rank within the classification called the infraorder. So they go from suborders to infraorders and then to superfamilies. Let's see. So the first group to branch off of the tree of life of the Diptera is something you've probably never heard of unless you come from out west in the mountains of the western US and western Canada. It's a group called the mountain midges, or the family Deuterophlebiidae. These are this is a very relict group of flies, very small. The adults are very short-lived. The larvae are aquatic and are typically found in the flat, let's see if I can actually say that, fast-flowing streams in the western United States. Let's take a look at one. This is an adult Deuterophlebiid. This weird forewing with this pleating in it and this big lobe at the base is very typical of them, as is the hugely enlarged mesoscutum that gives it kind of humpback appearance. This is a tiny little head down here with elongate antennae. And so this is a male. You can see the male genitalia up there. This is a mountain midge, the Deuterophlebeidae, or because they're in their own infraorder, this is the Deuterophlebiomorpha. The Diptera systemitist put the suffix morpha on their infraordinal taxa. Remember, infraorders not governed by the code, so there's no rules. They just decided that they'd add a little bit of consistency to help people recognize what the infraorders were. So the maple flowers are falling apparently into my water glass. It is water. So at the base of the dipteran tree of life, we have the Deuterophlebeiomorpha, and then we have a series of infraorders that kind of come off one after the other. The Tupulomorpha, which you're very familiar with, I'm sure, the crane flies, and I suppose I could have called them winter crane flies, although it's a different group. They're not true crane flies. And then the Psychotomorpha, which include moss flies, sand flies. Phlebotomus there is a genus of sand flies. Phantom crane flies. Many of these, maybe even the vast majority of the psychotomorphs, are totally harmless creatures, certainly as adults, and as larvae, they're just feeding on detritus and maybe some plant materials with aquatic immature stages here. But within them, within the Psychotomorph, and specifically within the family Psychotidae, which includes moss flies, we have the genus Phlebotomus, which is a vector of a protozoan called Leishmania. And this protozoan causes a disease called Leishmaniasis. I almost used the Portuguese name, Leishmaniosis. This parasitic protozoan, it's an intracellular parasite, can cause the destruction of cartilaginous tissues, and so it can be severely deforming if it gets into like the cartilage of your nose or the cartilage of your ears. But there are also forms that, if untreated, can be fatal. So this is a very, very serious disease, both aesthetically in terms of deformity, but also because it can easily be fatal. And I guess I didn't mention this, but of course the phlebotomus, the sandflies, transmit these parasites by taking a blood meal. The hematophagy, the feeding upon blood, has apparently evolved independently within the order Diptera. I have an accounting somewhere, but it's probably at least a half dozen times that blood feeding has evolved within the flies independently. And so this has been a useful evolving this habit has apparently been a useful thing for flies to gain nutrition. Typically, but not entirely, this is associated with females taking blood meals. There are a few groups of hematophagous flies in which the males also take blood meals. But back to these kind of ticking through these infraorders of true flies. The next one to come off is the Culicomorpha, which, as the name implies, you probably recognize culicids as the family of mosquitoes. But this also includes the chironomids and chaeoborids, various kinds of midges. There's a huge number of species in here. There's something like 3,500 species of mosquitoes, but that's dwarfed by the species richness in midges. Many of these, probably predominantly, we have, again, aquatic immature stages, although a number of midges can also be found in soils. And so we have both terrestrial and aquatic immature stages here. So just to take a quick look at some representatives of each of these infraorders, this is a classical crane fly of the infraorder Tupulomorpha. Very familiar to you, and of course everybody all of the news organizations, whenever there's an outbreak of dengue or some other mosquito-transmitted disease, they'll look for the most scary-looking mosquito they can find on the internet and end up with a species of crane fly and labeling it as a mosquito. This is a moth fly, the family Psychotidae. This guy is about 3 millimeters long. Maybe a real big one is 4 or 5 millimeters long. They get their name because of this dense hairy covering on the wings, reminiscent of the scales of a lepidopteran. The immature stages here are aquatic, and they're very common living in drains, feeding upon the organic debris in drains and any microorganisms that are living there. So it's not uncommon to see these flies associated with public restrooms. You'll see them sitting on the wall. They're perfectly harmless creatures, though. So that's the family Psychotidae. But in that same family, we have the sandflies, the phlebotomines. And here we have one that's taking obviously a blood meal from a human. Tiny, very fragile creatures, which their fragility belies the devastating impact that contracting in case of leishmaniasis can incur. Then we get into the Culicomorpha. So this is a chironomid, a male chironomid. You can tell by these just absolutely gorgeous, elaborate antennae. You're probably aware that many male mosquitoes use their antennae in order to locate mates by homing in on the sound produced by the beating wings of the female. So presumably these guys are doing something similar with these plumose antennae, feather-like antennae. This is the female, obviously very short antennae on here because she's not listening for the males. These things emerge in monstrous numbers. And one of the other interesting things about this is in some chironomids, the larvae actually have independently evolved hemoglobin in order to assist them in respiration and getting oxygen from the sediments, the aquatic sediments in which they live. We have hemoglobin occurring not only in mammals but in chironomids, and also there's a group of aquatic hemiptera, one of the back swimmers that are red and have independently evolved hemoglobin to assist in oxygen uptake from the water. And of course, a member of the family Culicida. I wanted to get the most elegant mosquito I could. So this is a species of the genus Sabethes, which is a neotropical species. The males use these paddles on their legs. They're not really paddles, they're just really elongate hairs in order to signal to females by waving the legs in the air in courtship, and the females respond to that thing. You can see these are so beautiful because they're metallic blue. These guys can transmit, the Sabethes that is, can also transmit some diseases, things like dengue and I think yellow fever and dengue they can transmit. But typically these mosquitoes are feeding upon monkeys. They're very, very, very shy, and you have to be extremely still in order for them to come in and feed on you. This was obviously taken in a lab where it's a little bit more controlled. So those 4 suborders, well, actually, I got to include one more here: the Bibionomorpha, which are the March flies. That refers to the month of March because the Bibionas are very typically come out in early spring. As a matter of fact, they should be flying right now. You should be able to find lots of March flies, even though it's April. and the family Cecidomyidae, which includes a huge number of gall flies, flies that cause galls on the growth of galls on plants, and then their larvae feed within those galls. And then the fungus gnats, thousands of species of fungus gnats in the family Mycetophilidae. There's some other families that go into this group as well, but these are the big boys. So these 4 infraorders pretty much comprise, maybe 5 if we include the Deuterophlebomorpha there, what classically would have been called the Nematosera. And instead of the Nematosera being a monophyletic group or a true clade, a true branch on the phylogenetic tree, it's more what's called a grade, a stepwise addition of apomorphic characters until we get to the sister group of the Bibionomorpha and get into the Brachycera. So probably the most infamous of the bibionids or the marsh flies are the so-called love bugs that are plague people who live down in Florida, because these things emerge in almost astronomical numbers sometimes from their aquatic the aquatic habitats where they live as larvae. And I mean, I've driven through love bug flights with the things being splattered on the car. And the thing is, you really need to wash your car down, because the hemolymph and the fat body and whatever else is on the insides of these creatures will take the paint, literally take the paint right off your car if you don't get rid of that stuff. Many bibionids have these very striking color patterns, suggesting, I don't know that anybody's actually ever tested this, that this is an aposematic coloration, and maybe the fact that their hemolymph will take the paint off your car means that birds and lizards and stuff shouldn't eat them. So that's their warning to stay away from me. But many of them are very colorful, even the ones up here. The Cecidomyidae, thousands of species of gall fly, or gall midges, I guess somebody corrected me once, that we should be calling these as common names. These are minute, extremely delicate creatures that occur in just a huge richness of species. Some of these are of agricultural importance because of the damage they do to the plants. For example, here's some of the galls that are caused by Cecidomyids, and so this takes away the energy from the plant. If you have a huge infestation, you wouldn't have normally developed leaves with many more of these gall flies developing in there. So we have gall flies or gall midges, and we have gall wasps. We have thrips that produce galls. All of these things are producing like these kind of little benign cancers on the plant. And then and gall aphids, I forgot to mention them too. And then the insects feeding on the inside. Well, that just brings up, there's also gall mites, right? So lots of organisms have developed this ability to take over the developmental mechanisms in the plant to produce these structures. And they feed inside, protected from their many of their natural enemies, not all, on the outside, and protected from the plant's defenses as well. A mycetophilid, again, a huge group. The larvae of these feed on fungi, the fruiting bodies of fungi. So if you find a mushroom in the forest and it's been there for a little while, then you almost certainly will find the larvae of mycetophilids. The name literally means mushroom-loving or fungus-loving flies living within in between the gills of those mushrooms. So if you're a mushroom hunter and you don't want that particular source of protein, perhaps you need to be careful and wash out those fly larvae that are living within. Okay, let's get down to the home stretch here. Brachycera, those guys with the very short antennae. There's still some phylogenetic work that needs to be done in order to nail these things down. But in the remember I contrasted Orthorhyncha, the straight seamed, versus the Cyclorapha, the curved seam flies. In the Orthorapha include the infraorders Tabanomorpha and Stratiomyomorpha, so the horse flies and deer flies, and then the soldier flies and the Stratiomyomorpha. And then a group is recognized at the superfamily level, the Acyloidea, the assassin flies, or the family Acylidae, and bee flies, the family Bombyliidae. Also in Brachycera, we have Empidoidea, the dance flies, so-called because they mate in these huge swarms, where actually they're not hand-to-hand dancing, but they're flying in these swarms. And well, I have a little bit of story about Empidids when I show you a picture of them. And then the Cyclorapha. So Cyclorapha is a subdivision of the Brachycera. And it's not just Orthorapha versus Cyclorapha. It's a little bit more complicated, the relationships here. But the Cyclorapha are characterized by a couple of things. First is that up to this point, the larval stages of these flies have had well-developed head capsules, a cranium, sclerotized cranium. In the Cyclorapha, that head capsule is lost, and so the flies, the maggots, are very membranous. Secondly, when these flies in the Cyclorapha go to pupate, they do not molt out of the last larval cuticle. Rather, they molt within it. The last larval cuticle becomes sclerotized and melanized, so hardened and darkened, and the pupa of the fly is actually found within. And so this sclerotized shell within which the pupa is found, which is actually the cuticle of the last larval instar, is called a puparium. So a couple of members of the Tabanomorpha. These are my most. I even dislike these guys more than I dislike mosquitoes. We have horseflies and deer flies. So this guy is probably only as long as the abdomen as horsefly, or even smaller. But these guys are characterized by being very strong flyers. Many hematophagous species, so many are taking blood meals, and did I say they're very strong flyers? That they will follow you as you walk through the forest. You can usually walk away from mosquitoes or something like that. But these guys are strong enough to keep up with you. And really worse than that is that they are so hard-bodied. You swat a mosquito, you kill it. You swat a deer fly, the deer fly looks back up at you and says, "Is that all you got?" And will fly away. So you really have to hit them hard in order to get rid of them, if you're fast enough to do it. This is a soldier fly in the family of Stratiomyidae, belonging to the infraorder Stratiomyomorpha, closely related to the horse flies and deer flies. Very characteristic antennae, this enlarged third segment of the fly. Some of these are quite large, and they're fairly common. They have both aquatic and terrestrial immature stages, where most horseflies, most tabanids, have aquatic larvae, although there are a few species that the larvae will live in the forest floor. But these guys have both aquatic and terrestrial immature stages. Let's see who's next. Yes, the dance flies. This is a female, yes, a female empidid. And as I said, they're called dance flies because of these elaborate courtship procedures that they go through. They're predatory as adults, as you can probably surmise from this beak that they have. But they have been very popular subjects of study because of I'm sure you would deal with this in your insect behavior course. They go through these series of evolutionary steps in courtship behavior, where sometimes a male in the the beginning stages, a male may capture an insect prey and present it to the female. Then the male in later stages, I mean, this is not an individual going through these stages, but evolutionary steps that people have kind of pieced together throughout the thousands of species of these flies. Males start to add glandular secretions to this prey item that they've captured to make it a ball and present it to the female. And then they go to the next step, and the males don't even bother to catch the insect prey in the first place. They just make the glandular secretion and present it to the female, and she accepts that. These so-called nuptial gifts are presumably a means of the female to assess the potential fitness of any offspring she might produce by that male. And so a male that's able to present a female with a big prey item or a big glandular secretion for her to feed on and gain energy from for her own production is more likely to be able to mate. So it's a very, I don't know all of the gory details about this the evolution of these nuptial gifts in flies, but it's a fascinating. What I do know, it's a very fascinating story. And so now we've come up to the Cyclorapha, excuse me, within which we have the Schizophora. Literally means split bearing. And we'll get to that in a second, but I've also already showed you a picture of a member of the superfamily Syrphoidea, the flower flies. The family Syrphidae is the most prominent and abundant group within the Syrphoidea. In the Schizophora, this split refers to the frontal suture, a suture on the frons of the animal, that it marks the position of the telinum. So the telinum is an eversible sac of the head that the fly uses. It inflates with hemolymph, so it contracts the muscles in the body, forces hemolymph into the head, pushes out this sac called the telinum. And it's with that sac that the fly pops the top on that puparium, that last larval instar cuticle within which it is pupated, in order to emerge. And so this is basically, remember, these guys are not going to have typically well-developed mandibles to chew out of through that cuticle as something like a moth. Well, the moth is going to digest it, isn't it? But some other organisms will. It uses the hydrostatic pressure produced and pressing with the telinum to pop the top. If you see a puparium from which a fly has emerged, it's like a very clean split, whereas like the cap has come off. Within the Schizophora, we're going to find two groups, and there's some dispute about monophyly here. The calyptrate flies are clearly a monophyletic group, and this includes the muscoid flies, the ones that are so familiar to you, house flies and blowflies and flesh flies and the like, but also a vast array of mostly quite minute flies. The bane of insect taxonomy classes. You can count yourself lucky that we didn't devote 2 weeks to you keying out calyptrate flies, because they are notoriously painful to identify. The identification relies upon the number, position, orientation of these bristles on the head and on the typically dorsum of the thorax, on the mesothorax, sometimes in the pleura as well. Calyptrates are clearly a monophyletic group. Acalyptrates, well, at least there's at least a big chunk of them that form a monophyletic group, but there's lots of leftovers where we don't really know how they fit in. So, starting with the flower flies again, this is the same, probably looks like even the same species that I illustrated the haltere with. But one thing I wanted to point out here, flower flies are well known, very, very well known for having many, Batesian mimics, that they are imitating typically, usually a hymenoptera, stinging hymenoptera. You know, is that a bee or is that a fly? Some of them are quite elaborate. There are some species that are just excellent mimics of bumblebees or honeybees and the like. And so, how do you distinguish these, especially when they're on the fly? Well, there's a couple of, well, not on the fly, but on the fly you would look for, do they have one or two wings, right? One of the characteristics of syrphids, the family Syrphidae, is something called a spurious vein. The word spurious simply means something that is not true, it's not genuine. So what we have is something that looks like a genuine wing vein, longitudinal vein of a wing, but is not, in fact. I've got a drawing here that's a little more clear in the next slide, but let me just point out the spurious vein on this guy. If you look right here, this follow this line right here, back toward the base, and see how it splits that cell there and continues up here. That's the spurious vein. That corresponds to no real longitudinal vein. It doesn't fall into the system of costa, subcosta, radius, media, cubitus, etc. It's a spurious vein. It's an independent evolutionary development in these flies, and it is one of the defining characteristics of this huge family of flies. Just to be a little bit clearer, so here we have a syrphid wing with all the veins labeled. Capital letters, I'm sure you remember the convention. Capital letters refer to the longitudinal veins. So here we have the part of the radius, actually the radial sector. Here we have the subcosta. Actually, this is not a vein, that's the pterostigma, so that blood cavity there. Costa, radius, medius, cubitus, fused with the anal vein, and then anal veins back here. The spurious vein is this guy here, SV. Rises here and runs, in the same kind of path that a longitudinal wing vein would, except it's between the radius and the media. The media kind of takes a contorted path here. And so this is an entirely new development, but characteristics of syrphids, a surefire identifier for this family. Okay, Cyclorapha. So when I've been talking about this frontal suture, here's what I'm talking about. It's this area here, kind of crescent-shaped area here, that goes over the top of the antennae. There's the antennae there, and down towards the oral cavity, which is down here. So this arc here is the frontal suture. So in an adult fly, this frontal suture is pretty much closed up. It's solid. You can, all the blood pressure, hydrostatic pressure that the fly can generate is not going to do anything that it's fused up. But when the fly is newly emerged from the pupal stage, this is the point where the talinum pops out. So I have a video, let's hope it works right, to show you the fly, a newly emerged flesh fly that is trying to get out of a petri dish actually, but it keeps expanding its talinum so you can see this structure. Get rid of my pointer. That didn't work. Let's see. There we go. See the talinum pop out there? So this is what it uses to pop the top of the puparium in order to emerge. I still have my laser pointer. So as I said, the Schizophora includes many very, very common groups. This fly is a member of the Calyptraes, and I told you the calypters are these large lobes at the hind margin of the wing. This is a calypter here, and you can see it over on this side too. If you pulled that wing out, it's going to be right toward the posterior articulation of the wing with the thorax. And these are acalyptrate flies. These are actually fairly large acalyptrate flies. There are a number of them that are more the size of a Drosophila. Again, notoriously difficult to identify. The taxonomy largely depending, you see these bristles, these black bristles on the thorax and on the head. The position of these, whether they're oriented, on the top of the head, or are they straight up and down, or do they cross over? Do they bend back? Do they bend forward? How many of them there are the characteristics that's used to identify these. Not helped at all by the fact that often these bristles can break off, but when they do, they leave an empty socket so you can tell where a bristle may have been in the past. So, my gosh, here we are. That's all I want to say about flies, except for the summary again. But I want to reminisce a little bit and basically ask myself and ask yourself, if we achieve the overall goals that I had set for this course from the very beginning. And my goals were not to make you learn hundreds of families, learn to be able to identify hundreds of families of insects just on sight. There are plenty of resources now more than ever, not only written keys, but lots of illustrative material available on the internet to help you with identification of groups. But I wanted to give you a sense of the scale and the makeup of insect diversity, the most diverse group of organisms, well, let's say animals on the planet. I guess I'll concede that maybe there's more bacteria than we have any know about. Wanted to give you some context to understand the evolutionary history of insects, both the absolute timing, but also the relative timing. What came first? Which things came together? Got more maple flowers falling. To be able to identify some of these things, at least the common families of insects, to show you some specimens, to give you firsthand experience with them. And then also to understand the principles that underlie the way we organize all our knowledge about insect diversity, but how phylogenetic inference actually is done, what the theory behind it is, what some of the pitfalls and what some of the practices are, and then also to be able to interpret those trees, to understand. As I said, evolution is not this scala naturae from the old days, which simply is a fancy Latin term meaning the ladder of nature. Evolution is not a linear process. It is an anastomosing process, a branching pattern. And sometimes, I guess if you have hybridization, those branches come back together, which makes things even harder. But so these are the kind of goals that I had from at a high level. So I hope that you come away from this course with a better understanding of what systematics is. It's not identification. Okay, and it's not only taxonomy that is putting things in a classification and putting names on things, but it's investigating the relationships among organisms, how they evolved, and the evidence involved in that. And so I think it's very important that entomologists understand what systematics is, even if you're not destined or predestined to be a systematist yourself. But give you that 30,000, I guess I'm saying the same thing over and over again, and give you 30,000 foot perspective on what insect diversity looks like today, to show you both the forest and the trees. And that last euphemism there, the gift that keeps on giving. There are so many species and so many different biologies that it's like a never-ending source of just fascinating stories and intriguing, compelling, obsessiveness worthy, if that's a word, problems for you to explore. So it's the gift that keeps on giving. There's always something new to discover. You never as a systematist, I never think that I have seen the be all and end all of what nature has to provide us. So I wanted to close. People have asked before, what's my favorite insect? And this, I have to say, is, I have lots of favorite insects, I must admit, but this is one of my favorite insects, both for the memories that it brings back. I first encountered, this is a phantom crane fly. Uh, It's a very distinctive species, Bidicomorpha clavipes. Bidicomorpha actually means in the form of Bidicus, and we saw Bidicus in the hanging flies, and you can kind of see why it would get that name. The specific epithet clavipes refers to these enlarged, pes means leg or foot literally, but it's referring to these fat segments here, so club foot segments. I started to say that the connotation that this animal brings to me is of spending summers in the Adirondack Mountains in northern New York. And when these guys emerge, I mean, they call them phantom crane flies for their common name for a reason, because as they're flying, this black and white pattern means that you might catch a glimpse of it, but then it almost literally disappears right in front of your eyes because they hold their legs widespread, kind of like this individual is sitting here. That's how they fly, and so they really kind of disappear. But I associate them with the coming of warm weather in the mountains and being by a lake, swimming, fishing, enjoying life. So that's got to be one of my all-time favorite insects. Not to mention it's beautiful, isn't it? So it's going to be impossible for me to do a summary in any depth on a single slide. So let's just summarize this last video at a very high level. The subject was Antliophora, which used to be composite, a monophyletic group comprised of three orders: the Mecoptera, the Diptera, and the Siphonaptera. Although now the Siphonaptera seem to, it's looking like they really ought to go into the Mecoptera and at best be considered just a suborder. The Antliophora is clearly monophyletic, but not for the reasons that we originally thought. That sperm pump, well, common within the group, appears to have evolved independently several times within the Antliophora. So the monophyly is really based upon the molecular evidence. The order Mecoptera is a relic group. I have a lot of fond memories of Mecoptera as well, but not many species, but quite a diverse biology, encompassing both predators, scavengers, herbivores, and if you include the Siphonaptera within the Mecoptera, then ectoparasites as well. So lots of things going on there. In the Diptera, a lot of those terms in the old classification you're going to run across. So commonly we'll hear people talking about Nematoceros diptera, referring to that old suborder Nematocera, which we now know is simply a paraphyletic group. So a lot of those old terms you're still going to hear, so I wanted to be sure and introduce them. But recognize that the modern classification is basically using the real skeleton of it are these infraorders, which I walked through some of the very common ones. And as I started out the whole discussion of flies with one of the most, if not the most, biologically diverse, species-rich groups of animals on the face of the planet that have a tremendous impact on human well-being and that of other animals through their unfortunate propensity to harbor microorganisms that cause a variety of diseases. So I think it's time to wrap this up. We're pulling up on an hour and a half, so this has been a little bit long-winded on my part. I hope that this whole experience has been useful to you and provide you context for understanding both trees as you see them in the literature, and the evolution, and provide that framework for understanding the evolution of insects writ large. So with that, I thank you for all of your attention, kind words, and I wish you the best, and hopefully I'll be seeing you in person in the very near future. Bye-bye.